greetings to you. Welcome to the Eastern New Mexico University Reads program, which I am given to understand that it's supposed to encourage young people to read more books and to support our libraries. I'm Dr. Manuel Varela. I'm professor of biology here at Eastern New Mexico University. The title of the talk today is Molecular Mechanisms of Bacterial Drug Resistance, the Antiporter Motif, and Inhibition of Multi-Drug Efflux Pumps. Don't worry, I will explain all of those things to you. But first, I want to tell you a little bit about myself for those of you who don't know me. I have a PhD in biomedical sciences with an emphasis in biochemistry and molecular biology from UNM, University of New Mexico in Albuquerque, the Health Sciences Center. I also have postdoctoral experience in microbial physiology at Harvard University in Cambridge and Boston, Massachusetts. And I've been a faculty member here at Eastern for about 23 years now. Right, so let's just get into it, shall we? All right, so antimicrobial agents are mechanisms that are meant to kill bacteria or inhibit their growth, okay? So we use them to treat infectious diseases. There's two types of antimicrobial agents, physical type and the chemical type. Physical type is just like UV light or radiation or heat or something like that, but chemical type might be antibiotics or disinfectants, such chemicals like that. These chemicals are present in everyday items like, toothpastes, mouthwashes, apparel. They're in cattle feeds. They are widespread. And because of that, they enjoy widespread use. This widespread use encourages the growth of bacteria that are intrinsically resistant to those chemicals and result in what we call drug resistance. So there are several mechanisms of bacterial drug resistance. One of them, number one, is called blocking the entry. Now here the antibiotic cannot get inside a bacterial organism to inhibit its growth. Okay, so they are prevented from gaining entry. A second mechanism, number two here, is called inactivation of enzymes or by enzymes. Here, an antibiotic will be bound by an enzyme and then chemically processed to make it not work, or what we call inactivated. A third way would be to take a target, here is a target of an antibiotic, and then mutate it such that it no longer binds to this target, and the antibiotic can't do its inhibitory or killing function. The one that I'm most interested in is one called efflux of antibiotics. Here, the antibiotic will go into a cell, but then quickly be exported to the extracellular part of the cell where there are no targets. Okay, so these are four basic mechanisms of resistance to drugs. Okay, if you use a single drug, that would select for a bacterial variant that is actually multi-drug resistant, and these multi-drug resistances compromise the treatment efficacy of infectious diseases caused by those drug-resistant microbes. Another problem is that these mechanisms are poorly understood. Now, I mentioned one of these mechanisms earlier, the efflux of antibiotics, and they are poorly understood in terms of how they work and how they're shaped. So they belong to a larger class of proteins called solute transporters. So let's talk a little bit about solute transporters. These are proteins that sit in the membrane of the cells of all living creatures on Earth. And because of that, they are essential for all life on Earth. One problem with that, though, is that many of these solute transporters are defective in genetic diseases. Adrenoleukodystrophy, cystic fibrosis, and the like. And again, they are poorly understood. So we kind of look at them like a dark box in the membrane, and we don't really know how they work or what they're shaped like. And if we knew what their shapes were, we might be able to inhibit them. Okay, so if we took a look at a bacterial cell, here's a membrane. There are several types of solute transporters. The first type is called the primary, and they use ATP to pump protons out, or substrate out, or a substrate in some cases. Another case is called the phosphotransferase system, also known as group translocation, where they'll take a substrate, like a sugar for instance, and bring it into a cell and then put a phosphate onto it. And that'll lock in that sugar into a cell. Another type of transport system is the one that I'm interested in. It's called secondary transport systems, and they function by using a proton gradient or a sodium gradient or an ion gradient as energy for them. And here's a gradient of protons, and there's three kinds of these secondary transport systems. One's called an antiport, where a product will be pushed out, and the ion that drives it will be pushed the other way. Another one is called uniport, where we only have one substrate, and the concentration difference of the substrate drives the uniport. Symport, both the ion and the substrate go in the same direction across the membrane. Okay. Now, when I was in graduate school, I tested the hypothesis that sequence variations account for differences in transporter function. Now, I study in particular a large class of proteins called the major facilitator superfamily. Sorry for the big long name. I didn't name it, but somebody else did. But they harbor in this family symporters, uniporters, antiporters that I had just mentioned earlier. They have a diverse array of solutes. Oh, I don't know, Krebs cycle intermediates, sugars, amino acids, antibiotics, ions, small molecules, water even. And they use different modes of energy to drive them. And we think they have similar protein structures, and they share similarities in their amino acid sequences. In particular, some of these transporters have conserved amino acid sequence motifs. This means that a certain type of amino acid sequences are present in all of them, or maybe in certain functionally related subsets of them. Okay, so my focus was on these highly conserved sequence motifs. So to test this hypothesis, I was going to clone a gene mediating the efflux of an antibiotic, tetracycline, sequence it, compare that sequence with all sequences that are known in the database, mutate the most highly conserved residues of amino acid sequences, and then create a structural model that might help me to inhibit those transporters. If you inhibit the transporters, you might inhibit antibiotic resistance. Okay, so first we clone the gene. Here's a gene for tetracycline resistance, cloned into a vector and put into bacteria. I asked the computer to deduce the amino acid sequences alongside the protein sequences. And then I asked the computer to compare those deduced amino acid sequences with what was at the time known about other sequences, and then do a statistical analysis of the validity of these comparisons. Then I asked the computer to design a phylogenetic relationship between the one that I'd sequenced with other sequences, and we found that our sequence was highly related, homologous if you will, to multi-drug efflux pumps. Now another group in Australia discovered that there was if you ask the computer to take a sequence and do something with it two-dimensionally, it shows that the protein goes into the membrane, in and out about 12 times, sometimes more. And in the middle of this block here, we call it alpha helix, this motif was found. And here's the alignments of amino acids. They have glycines and a proline in this helix, number 5. And it's present in antiporters. So when I was in graduate school, I decided to name it the antiporter motif. Okay, we also found that this antiporter motif is missing in symporters and uniporters, but present in antiporters. This is one of my favorite scientists, Linus Pauling. He's famous for vitamin C and treatment of infectious diseases like cancer. And he's the one who discovered the alpha helix. And it looks like a ribbon structure. Here's a space filling model. and sometimes we just envisage them as a like a cylinder. Okay, so I decided to model that fifth helix of these antiporters, and here is one of them, and this is what this looks like if it were unmutated. Okay, and every round circle here represents an atom. And I decided to take those bacteria and mutate their tetracycline efflux pumps such that this highly conserved glycine here, which has the side chain of a hydrogen, mutated to an alanine, and then measure its function, and we lost about 74% of its function. And then, if you watch here closely, as I put on the next slide, watch this part right here as I make one more amino acid change. Okay, ready? See that? See how much bigger it got? And that is a subtle difference, okay, in structure. But it had a profound effect on function in that it lost almost all of it. When I replaced all the other 19 amino acids in that one position, all activity was lost. So we inhibited tetracycline antibiotic resistance with a minor change. Okay. Another piece of work we did was a molecular mechanics modeling. We asked the computer, make a structure that uses a minimal amount of energy to make it. So I took this helix found in antiporters and asked the computer to energy minimize that structure, and we found that it was bent or kinked. And where in symporters and symporters and uniporters, where the antiporter motif is missing, the kinking, the bending does not happen. Okay, but only in the antiporter motif. Right, we're going to skip ahead to the year 2013, where another group based in Israel and Germany tested that antiporter motif, and they showed my helix to be bent, helix number 5 to be bent. And now they said that this helix forms part of a so-called molecular hinge region, in that when the protein works, it hinges. Okay, it works on a hinge. Okay, all right. Another group based in Germany and Japan took this motif and they confirmed the bent nature of the helix. It's shown here in green. And this is published in Nature in 2018. They also show that this helix turns around or twists. Okay, so unfortunately, this antiporter motif is both twisted and kinked. All right. The antiporter motif started generating a lot of attention in terms of mutagenic analyses of other amino acids in the motif and in supporting structural as well as functional roles of the motif. So this is what we knew when I started working in this area, basically an amorphous set of a two-dimensional structure. And so what I decided to do was just start collecting mutants that would affect their function, because I already knew that subtle differences affected their function. So I started putting little dots in locations where mutations affected their activities. And here's one activity. If the wild type has this much activity, one mutation would lose function here or lose a lot more function there. So I was actually not only losing function, but changing function. And as I gathered these locations in the proteins that made more mutations, I would try to coalesce them into a three-dimensional structure by orienting all the amino acids into one little domain to make them more of a functional structure. All right, so at about this time, one of the proteins that I worked on called lactose permease, and these are amino acids that I showed in these studies to be important in binding a substrate like a sugar or maybe a proton, or work in the transport cycle. This study, published in Science in Ron Kabach's laboratory, showed that my amino acids constituted key substrate binding sites, proton binding sites, right there, right here, and we're making salt bridges with key structural modulators. Okay. So as a professor here at Eastern New Mexico University, we decided to play around a little bit with more sugar transport studies. And I decided to take a transporter called CSEB for the chromosomally encoded sucrose carrier protein B, sorry for the long names, and mutate it so that it transported, instead of sucrose, maybe maltose. Now maltose makes malts taste great. Sucrose makes almost everything else taste great. So if I could mutate them to have, acquire differences in sugar specificities, that would be a good thing. So we decided that to put it on plates that would turn red if they mutated to transport maltose. And unfortunately or unfortunately, we found out that it already turned pink when we put it on maltose. So maltose was already then a substrate. And that is why we hypothesized that it was a substrate. And if it's true, then we could measure maltose transport as a function of time in addition to sucrose. And yes, we got a positive signal, therefore it is a transporter of maltose. Being a biochemist, I decided that I should like to look at kinetics, Km, Michaelis constants, maximum velocity of transport across the membrane. And we got positive signals for both sucrose and maltose. If it's an active transporter, then it'll show what is called an uphill accumulation of the sugars, and maltose is actively accumulated, maybe even more so than the sucrose. All right. Now, when we sent this to be published, the editor came back and said, well, can you block the binding sites with the other substrate? And that was a good question to ask. So we added more maltose, and we were able to block the transport of sucrose. Alternatively, we added more sucrose, and were able to block the transport of maltose. So they blocked the transport of each other. All right, so we decided then that sucrose and maltose share the same binding sites. And we published that work in 2009. Okay, so cholera is an important disease, okay? And we think that we are in the 8th pandemic of cholera. Now, the 7th pandemic ended somewhere in the 60s, but cholera is a serious disease, right? So we figured that if we could learn more about these antibiotic resistance mechanisms in cholera-producing bacteria, we might be able to modulate this activity to inhibit drug resistance in cholera. So we decided to test the hypothesis that a genetic determinant called EMRD3 from Vibrio cholerae, the causative agent of cholera, is a multi-drug efflux pump. All right. Now, as I said earlier, these mechanisms are kind of poorly understood, but they would, if we know more about them, they would make good therapeutic targets and maybe maintain therapy or restore therapy. The efficacy of treatment of therapy in antibiotics that have been compromised by resistance. All right, so we cloned its gene. We did a bioinformatical workup in which we compared our sequence with those of known sequences and found that it is homologous to many other unknown sequences. Excuse me. All right. We asked the computer to make a two-dimensional model, and it shows 12 helices and the antiporter motif in helix 5. We did a physiological study, or more of a microbiological study, in which we asked the question, how much drug does it take to inhibit the pumps? And we found that it takes a lot of antibiotic, and the gene makes it takes a lot of antibiotic to kill it compared to cells that don't have the gene, and it conferred resistance to multiple antimicrobial agents by many-fold. Okay, so we did another physiological workup in which we asked the cells to see how much drug they would accumulate if they had the pump or if they didn't have the pump. And if they had the pump, they would accumulate a lot less drug compared to cells that don't have the pump, where they accumulate a lot of drug, meaning that they export that drug outside of cells. we could do another experiment in which we load the cell with drug and then ask the question, if once you turn on metabolism, how much drug gets lost or pumped out in cells that have the pump versus those that don't. And we saw that there was an active transport mechanism at play. You can ask another question, if you add a proton, would it make drug transport better? And the answer is yes. If we add linezolid, erythromycin, chloramphenicol, protons get pumped out, okay, compared to controls, maybe not, protons don't get pumped out so much. And it happens with all three of these drugs that we tested. Okay. Now another question is, if indeed protons are important for the function, then if you change the concentration of the protons, you might modulate the activity. So if as we add less protons or more protons, activities go lower or higher, respectively, depending upon the nature of the drug. Okay, so now we know that it's an active transport protein that is run by protons. We published this work in 2009. Okay, we think they would make a great candidate for targets by antimicrobial agents in the future. Right. Another study we did happened to be involved with a salmonella outbreak in New Mexico and Texas. It was a short-lived outbreak, but it was extensive. But we noticed that it didn't last very long. So it suggested to us that either those causative agents of Salmonella enterica were either susceptible to medicines or they were not very pathogenic, that is to say, avirulent. So we did one experiment in which we tested the number of genes that confer virulence, and all the isolates had them. So these were pathogenic clinical isolates. These are all from New Mexico, by the way. And we also found out that they are susceptible to many antimicrobial agents. One exception was number 6, New Mexico isolate typhimurium serotype, number 6, that was resistant to ampicillin, cephalotin, gentamicin, chloramphenicol, amoxicillin clavulanate, and maybe not so much tetracycline, but certainly resistant to those. It was a known pathogen. Right, so this number 6 then is, we already knew about it. We confirmed its identity with the phage typing. The other ones were mainly sensitive to antimicrobial agents. All right, so we published that in 2010, having to do with New Mexico, by the way, and Texas. Okay, another study we did was we discovered a multi-drug efflux pump called LMRS from methicillin-resistant Staphylococcus aureus, also known as MRSA. MRSA is a serious pathogen. It and all other superbug pathogens are responsible for about 700,000 deaths of people per year. Every year we have that many deaths due to bacteria like this one, the MRSA infection. We cloned the MRSA gene and did a bioinformatical workup and found that it has 14 alpha helices and our antiporter motif in helix number 5. We did a multiple sequence alignment and found that it is highly conserved with many other bacteria, that some of them can be serious pathogens in their own right. Excuse me, like Listeria monocytogenes is a serious pathogen. We did a microbiological workup, and we found that cells that have the pump confer resistance to many antimicrobial agents compared to cells that don't have the pump. Okay. We did a physiological workup and measured transport, and we saw an efflux pump assay, in which we load the cells and the antibiotics gets pushed out compared to control. All right, we added drugs and measured protons and saw a positive signal of proton transport for 4 completely different antibiotics. So it is a proton-driven multi-drug efflux pump. We published this in 2010. Okay. Another study we did, and I'm kind of proud of this one, is that we discovered an operon. An operon is a genetic element on a chromosome with a collection of genes on it, and all the genes are devoted to one function. And in this particular case, it was mannitol utilization. Now, mannitol is a sugar with an alcohol group on it, and it's used for metabolism for cholera to grow. And this is a serious pathogen, 0395, a pandemic kind, causing kind of pathogen. So we, here's the operon in its theoretical structure, its gene structure. We focused on gene number 1, MTLA, and hypothesized that it's a PTS transporter. So we saw positive fermentation of mannitol in cells that have the pump versus those that don't have the pump. We did a bioinformatical workup and found that it is highly homologous in the membrane to the PTS transporter systems. So we measured transport and saw a positive signal in kinetics with cells that have the pump compared to those that don't have the pump. And we published that functional data, and we moved on to the next genetic element. In this particular paper, and tested the hypothesis that it's a dehydrogenase enzyme. So we sequenced it, compared that sequence with others, and found that they are homologous to many other dehydrogenases. Its sequence shows that it's got conserved sequence motifs indicative of dehydrogenases. And I was coaxed to purify the protein. I don't like normally purifying proteins, but they said, well, we can do it, Dr. Varela. And I said, well, okay, give it your best shot. And they did. So they purified the protein, and there it is, nicely purified. I'll skip the background here. So if it's an enzyme, then we should be able to measure kinetics of it, and sure enough, we did. And when we tried to publish it, the reviewers came back and says, well, what's the effects on pH? So we did an effect on pH. What's the effects on growth or on temperature? So we measured the temperature. And what's the effects on growth? And we measured growth, and growth. I'll just skip a lot of growth. Now, we didn't have to be coaxed to say, well, if it's an operon and if the mannitol is inducing it, then the mannitol should induce the gene expression of those genes in that operon. And so the answer is yes, it does. And here is the induction of messenger RNA gene expression in a good cholera and a bad cholera and a non-pathogenic cholera. All right, so then we published the second genetic element, and we discovered an operon, which is kind of cool because I really love operons. Right, another study was to do an entire genomic sequence of one of the cholera. Now this one is a non-O1, non-toxigenic, non-pathogenic cholera, isolated from Puget Sound, number 15. It's just out there in the environment. And we did the entire genomic sequence in collaboration with the National Center for Genomic Resources in Santa Fe, and found out that it has 3,512 open reading frames, possibly genes, and we figured that it might have genes that are missing in it that are present in pathogenic cholera. 430 genes are hypothetical in that we don't know what their functions are. Okay, but the reason we did this was to test this hypothesis that pathogenic versus non-pathogenic cholera have distinctive genomic elements that are responsible for conferring pathogenesis or virulence. So we would test that by comparing the sequences of the non-pathogenic versus pathogenic. Excuse me, I'm losing my voice. Right, so our gene is non-pathogenic here and is distantly related to a cluster of pathogenic genes or genomes. If we compare one of the most pathogenic genes with our non-pathogenic gene, the pathogenic gene in red and yellow all around are compared with our good one in blue all around. You notice gaps. where the good one, the non-toxigenic, is missing genes dispersed throughout the genome. All right. So we looked at those regions and found that they confer a variety of cellular functions. One of them is, antimicrobial resistance, or cholera toxin making, or just growing, or waiting around to wreak havoc disease-wise. Okay, so I mentioned that if we could inhibit the efflux pumps, then we might be able to inhibit drug resistance. And we decided that many of the efflux pumps inhibitors are toxic, and so we need non-toxic efflux pumps. We decided that efflux pumps that are inhibitors that are plant-based, food-based, might be good non-toxic inhibitors. One of them was cumin, which makes foods taste great. I know people like cumin. And so we asked the question, does cuminaldehyde, its bioactive principle, cumin seed oil, do they inhibit the growth of cells? And the answer is yes, they do. Okay. Does it inhibit our pump? The question is yes, it does. And if you add increasing amounts of cumin, you reverse the accumulation of the loss of accumulation of cells, and so you inhibit efflux by the LMRS in MRSA cells. All right, so we did a bioinformatical workup, asked the question, does the cumin bind LMRS? And the answer is yes, it does, possibly at valine 371. Another study we did was measuring the effects of garlic on cells. This one having to do with cholera, and we tested two different hypotheses. One was that the garlic works together with other antibiotics to inhibit the growth better together than differently. And this meant that this type of interaction is what we call synergistic. Okay. The second kind of, the second hypothesis was that the bioactive principle of garlic, called allyl sulfide, inhibits our EMRD3 efflux pump. All right, so we found out that the garlic extract and its bioactive principle inhibit the growth of the good cholera, the bad cholera, and cells harboring and lacking our EMRD3 multi-drug efflux pump. If you add garlic to antibiotics, to cells that don't have the pump, or to cells that do have the pump, you enhance the efficacy of those drugs against these cells, both of these cells. Okay, all right. Now, when you measure synergy, you might find good combinations of these drugs that would work better together than separately. And we found that garlic synergizes with chloramphenicol, a good antibiotic. S means synergy, N means no synergy. We found synergy with erythromycin and garlic. We found synergy with kanamycin and garlic. We found synergy with linezolid and garlic, and maybe not so much with tetracycline and garlic. Vancomycin and garlic had a good positive combination. All right, we published this work in 2017. This work is not published yet. It's just fresh off the press. It was done just last semester by one of my graduate students. And she found out that one of our colleagues, Dr. Cho's trigonelone, inhibits our drug efflux pump in the MRSA. Cells that don't have the trigonelone, or the trigonelline, accumulate this much. Cells that don't have the pump accumulate a lot more, and cells that do have the pump but have trigonelline are blocked and accumulate a lot more. Okay. Another study that's not published yet is to ask the question if the single drug efflux pumps are related to the multi-drug efflux pumps can you convert a single to a multi-drug efflux pump by a subtle mutation? And the question is yes. And so we asked the question, if you put cells with tetracycline efflux pumps on a plate containing norfloxacin, can you find mutants that would grow that would be multi-drug resistant? And the answer is yes, you can. Because remember, subtle changes in sequence or structure would have profound changes in function. And that's just the way nature is. We found hundreds of mutants that did this. We sequenced about 10 of them and found that 2 of them had mutations in motif C, our antiporter motif, and another motif called motif A, doesn't have a name yet, that forms a gate. Motif A forms a gate, and motif C forms the hinge. Okay, so for future work, we're going to continue with our synergistic studies. We're going to continue with our modulatory studies, trying to inhibit ARC pumps. We're going to look for more targets with genomics. Okay, and those are the 3 focuses of study. And if you're interested in doing this kind of work, come visit me, and we will put you to work in the lab at Eastern New Mexico University doing this kind of stuff. Okay, while I have the floor, I want to brag a little bit because this is a library. We are in a library. I published my first book with a collaborator, Dr. Shaughnessy, Mike Shaughnessy, at a Christmas party. I told him how I liked his books. And he said, well, we can write one together. And I said, well, okay. And so we did. And this book is called The Inventions and Discoveries of the World's Most Famous Scientists, because in lectures, I talk about famous scientists, what they did, why that's important, all the time. So we put that into a book. We have another book that came out in 2019 about famous microbiologists. This is also in the library. We have a book that came out last semester about famous biomedical scientists and their discoveries, and that's soon to be present in a library or a bookstore near you. And then I'm really proud about this one because it's the first time that my wife, my lovely wife, Ann Higgins Varela, and I, and Dr. Shaughnessy, collaborated on a book together. This one's about famous biochemists. So this one's already out. I haven't actually seen it yet, but it should be available in the library this month. Okay. Right, so again, these are all the people that did most of the work that I described, okay? And they are named here, and collaborators and funding sources. And I think that would be it. Right, so thank you so much. I am so happy that you watched this video. And please do contact me by email, manuel.varela@enmu.edu, if you have a question, and I will be glad to answer them. So take care. Thank you so much. Have a great day.